Okay, shall we start? And yeah, thank you very much for everybody for coming this seminar, webinar series. This is a July webinar organized by GO2NE, Global Ocean Oxygen Network. And I'm very glad to have two outstanding speakers, Huang and Curtis Deutsch, today talking about temperature, oxygen in deep time and the marine world. And before starting the seminar, one technical announcement. And you may like to have a look at the first edition of GO2NE newsletter, Good newsletter. It's just out. And yeah, it's exciting newsletter, and you may like to have a look. And then it's a weekly webinar. So we have June, July, and this one is July one, and we are working on the next one in August. So please stay tuned. And yeah, let's start. And I'm really excited to introduce Dr. Huang, and in the Smithsonian Institution. She did a PhD in University of Hong Kong several years ago, and she's now postdoctoral fellow in the Department of Paleobiology in Smithsonian Natural History Museum. She's paleobiologist and micropaleontologist. She has been working on Cenozoic climatic change and Cenozoic climatic impact on shallow marine and deep sea ecosystems. So yeah, please join me to welcome May virtually if possible. And shall we start? May I ask you to start?
Okay.
I may
Thank
start.
you, Moriaki. Oh.
Yeah.
You can see my screen. Thank you. Thank you, Moriaki, for a nice introduction, and thank you for inviting me to give this talk. The host of this seminar, GO2NE, Global Ocean Oxygen Network, was initiated because of the alarming data that shows declining dissolved oxygen in the world ocean. This is a figure from the latest IPCC report. It shows estimates of marine heat waves, dissolved oxygen, AMOC capacity, and pH in the ocean today. Earth system is very complicated, and the changes in these parameters are likely interrelated. Let's look at the plot of dissolved oxygen. We see again declining values over time in the past observations. So these figures are an overall view of the average changes in the world ocean, and they show us consistent patterns among them. And so it is very insightful to think about. It is also very important to have comprehensive understanding on different ecosystems. Many researchers have been working on different ecosystems and in different local systems, such as coastal waters, open oceans, continental shelves, and enclosed seas. In particular, they're trying to understand and predict biotic responses to deoxygenation. The paper on the left-hand side was published by Peter, a colleague, and the GO2NE group last year. I was also a co-author of this paper. So today I would like to share with you my study in the Sea of Japan, a semi-enclosed marginal sea. The Sea of Japan is a marginal sea between Japan, Russia, and Korea. Today is well oxygenated, as shown by the relatively warm color in this figure of dissolved oxygen. However, it is not always the case, and I will show you in the rest of the talk that the Sea of Japan experienced quasi-periodic anoxic events in the past over and over. We will travel into the past 2 million years, or the Quaternary period, or the Pleistocene and Holocene epochs. The scientific objective is to understand what were the impacts of long-term climate change and oceanic deoxygenation on the benthic faunal variability. This talk focuses on deep sea ecosystem, Pleistocene climate events over the past 2 million years, and orbital scale deoxygenation events. The hypothesis is that the deep sea faunal variability was primarily controlled by the secular scale global climate events and secondarily by orbital scale oxygen fluctuations. Secular and orbital scale global climates refer to the natural climate variability that was essentially controlled by the orbit geometry of the Earth, including the shape of the orbits, the tilts, and the wobble. This is a nice diagram from Hodell's paper in Science in 2016. This diagram explains the concept of how the inputs, how the orbit geometry of Earth as an input to the Earth's climate system results in the tempo of the glacial and interglacial cycles that we observe. And this global climate variability is recorded by the deep sea, by the oxygen isotopic values of deep sea forms, as seen here, the famous Lysacki-Ramo LR04 record. We see on the left-hand side figure, there are numerous marine isotopic stages colored as gray and white background that shows the alternation between glacial and interglacial cycles. In the Pleistocene, there are two important secular scale climate events. The first is the mid-Pleistocene transition. It is fundamental shift in the climate cycles from a transition from lower amplitude 41,000 year cycles to higher amplitude 100,000 year cycles. So mid-Brujas events is the related events where we see the 100,000 year cycle further amplified, and at the same time we observe enhanced Arctic amplification, which is a very important topic right now. So these events had significant influence on the changes of ocean circulation, sea ice variability, ocean temperature, etc., and all of which influenced oxygen supply in the deep sea. The Sea of Japan is an ideal natural laboratory for studying the biotic responses to these climate events. Since about 1.7 million years ago, the Tsushima Strait in the south subsided, and the Tsushima warm currents has become the only major saline water influx to this marginal sea. During interglacial or high sea level periods, like today, the Tsushima warm currents flow from the south to the north and sinks to form the deep water. The open ocean convection and sea ice formation in the north brings oxygen to the deep water and ventilates the marginal sea. However, during glacial low sea level stands, because of these shallow straits that connect the Sea of Japan to its adjacent waters, the influx of Tsushima warm current was largely reduced during the low sea level stands. And the reduced influx of saline water has been thought to cause strong oceanic stratification in this marginal sea, and subsequently severe basin-wide deoxygenation. This deoxygenation event was recorded by the sediments. This dark sediments you see in the figure here was deposited under anaerobic conditions or eugenic conditions, especially in these thick dark layers that are characterized by high organic contents and sulfur. So the idea is to reconstruct biotic responses to these deoxygenation events using microfossils from marine sediment cores. Deep sea drilling programs such as DSDP, ODP, ILDP have drilled all over the world, and studies based on these cores have contributed tremendously to our understanding of the past climate change. In 2013, ILDP Expedition 346 revisited the Sea of Japan, and I was a shore-based scientist of this expedition. I made a lot of friends over this project during my PhD, and all of these wonderful scientists and technicians of this expedition contributed to the study I am presenting here. So the expedition was very successful with high recovery rates and high quality sediment cores. They mostly used the advanced piston coring to produce higher quality than the traditional rotary coring for the soft sediments here. And this high quality cores allowed the highest resolution sampling of my study. So I studied two sites that were drilled during this expedition. Both sites are located in the southern Sea of Japan, and you can see in the right-hand side figure of depth temperature profile that both sites are located in the Japan Sea proper water, or the colder, deeper water with overlying Tsushima warm currents in the surface water layer. This one side is deeper at 903 meter water depth, and the other side is shallower at 330 meter water depth. The deeper side has a longer 2 million year record with about 4,000 year time resolution of sampling. The other side has about 0.7 million year record with higher time resolution of 1,500 years of sampling. I used age depth model to calculate the age of each sample. In general, the deeper the sample is, the older it is. And the age model was based on biostratigraphy of foraminifera, radiolarians, and nanofossils. Tephra stratigraphy and stratigraphic correlation were also used to construct the age depth model of my sites. And to reconstruct biotic responses, I studied microfossils in the sediment cores. Ostracods are a group of excellent microfossils. They are crustaceans. They're tiny, with adult size ranging between 0.5 to 2 millimeters. They have calcitic shells that can be preserved as fossils in the sediments. And they're the only metazoan that have abundant fossil record in the deep sea. Just a quick recap that we are interested in reconstructing biotic responses to secular scale global climate events and orbital scale oxygen fluctuations. So I picked ostracods from the sediment cores. I identified them and counted them, and look at the relative abundances of each species through time. And I used color of sediment as the surrogates or proxy of oxygen levels in this study. The color was measured by an index called L star. So if it's completely black, L star value would be 0. If it's completely white, it's 100. In total, I have over 9,900 specimens, and 55 species were identified. These are SEM images of some of the major species in my samples. Here is the results. It's a non-metric multi-dimensional scaling of the assemblages. It's an ordination method that preserved the relationship between the assemblages in a two-dimensional plot. The dots are the samples, and in general, the samples are with more similar species compositions are closer to each other on these plots. And so the open circles are the samples from before the Mead-Broughas events. The light blue circles are from after the Mead-Broughas events, and the triangles are the samples during the Mead-Broughas events. And we can see on this plot that samples or the assemblages before and after the Mead-Broughas events are well separated. And this means there was a faunal turnover at the Mead-Broughas events. And this turnover was driven by a transition from assemblages dominated by a certain polar species, Acanthoceteres dunelmensis, to Ropersonites hanai, which is a Japanese endemic cool water species. These are figures showing the relative abundance of these two species over time. And before the Mead-Broughas events, we see higher abundances of Acanthoceteres dunelmensis, and after the Mead-Broughas events, higher relative abundances of Ropersonites hanai. After the Mead-Broughas event, there were amplified glacial interglacial cycles. So we interpreted that this transition corresponds to a warmer interglacial after the Mead-Broughas events, and probably there were enhanced Tsushima warm current influx during the stronger interglacials. Now let's look at the orbital scale variability. The left-hand side plot is the sea level over time over the past 700,000 years, or the 17 marine isotope stages. The right-hand side plot is the logged total abundance of during this ice age terminations 1, 2, 4, 5, and 7, where the sea level dropped more than 90 meters. And so there was recursive local extarvation or mortality events over and over in response to the deoxygenation. Phasing out of the mortality events, we do see recovery of ostracod communities. This is relative abundance of 2 indicator species, and I colored the background. The gray background represent glacial periods, and during these periods we see the dominance of Cridae sawanensis, which indicates a moderate oxygen depletion. They're infaunal, and they have a tolerance against moderate oxygen depletion. And in the white background, which are the interglacial periods, we see the dominance of Sideropteron hyalinosa, which is the species that indicate well-oxygenated environments. And so over time, we see the alternation between these 2 species. One indicates phasing in and out of mortality events when the oxygen was moderately depleted. And in the interglacial period, high sea level periods, we see more species that like well-oxygenated environments. So in Cascade multivariate regression tree, which is constrained clustering, age is the primary explanatory factor, explain about 28% of the faunal variability, and it identified 2 stratigraphic thresholds that divided the samples, and it identified the 2 important events in the Sea of Japan. One is the onset of the Tsushima 1 current at 1.7 million years ago, and the second is the mid-Brujas events at about 0.4 million years ago. And the color L star as a proxy of oxygen levels explain about 24% of the faunal variability. In total, about 50% of the faunal variability were explained by just these 2 factors. To summarize, we see local extinctions occur in response to deoxygenation. During eustatic sea level low stand, we see strongly stratified marginal sea with probably hypoxic condition in relatively shallow area, and eugenic in sites that are deeper than 900 meters. And in interglacial high sea level period, like today, the marginal sea is ventilated. And both sides would be well oxygenated. So the orbital scales, climate's variability govern the deep sea faunal variability. And after the mid-Brujas event, we see some fundamental changes in climate, and in response, there was also a faunal turnover in the Sea of Japan from more circumpolar dominated assemblages to assemblages dominated by cool water species that are endemic to the Sea of Japan. And of course, there's more work is required, and we're also interested in perhaps higher resolution or like centennial or millennial climate events that are possibly more relevant to what we want to know for the crisis today. Finally, looking elsewhere, we also see other studies based on microfossils that show global impacts of mid-Pleistocene transition and mid-Brujas events. The plots here are relative abundances of indicator species or group. The red plots show data from Kawagata and colleagues. They show that there was a global mass extinction of a particular group of benthic forams at the mid-Pleistocene transition. And in the Arctic Ocean, Cronin and colleagues show that there was also a faunal turnover of ostracods in the Arctic Ocean. And this would be another talk, but I just want to show here that there's this work. We're just beginning to understand the impact of these global events, and there is more work to be done. So in conclusion, microfossils are a powerful tool, and the talk I presented here shows high sensitivity of deep sea ecosystem and the interplay of global climate change, sea ice formation, regional currents, ocean circulation are very complicated. And under the ongoing warming, we want to be more knowledgeable about how these things interplay, and especially in the context of the Sea of Japan, the sea ice formation and the cooling of the surface water is really important to the ventilation and the oxygen supply in deep water. And ongoing warming has already, we already see observations that the warming of the sea surface water has reduced the dissolved oxygen in the deeper zone. So it is important to look deeper into these topics. And so finally, thank you everyone for coming to my talk. And I'd like to thank my collaborators, Moriaki, Carlos, Maria, Angela, Takuya, Tatsuhiko, Katsura, and everybody on board of Expedition 346, and the technicians in the University of Hong Kong and IODP.
Yeah, thank you very much, May. Great talk. So yeah, for the first question, I'd like to invite Curtis to say your comments or question to start Q&A session, and then I will pick Q&A online from Q&A chat box. Please fill in.
Hello, everyone. That was wonderful, May. Thank you. And thanks for the opportunity to start off the questions. I was wondering if you have thoughts about why the Sea of Japan seems to, it sort of goes in a counterintuitive direction during the interglacial transitions, where it becomes anoxic during the colder intervals, the glacials. I was wondering if you have any thoughts about either the biological response to that or the causes of it, perhaps.
So I guess your point is, if it's colder, there should be more sea ice formation and sea surface cooling that brings oxygen to the deep water. First thing is, the Sea of Japan is really well studied, and every paper I see by the other researchers and pioneering researchers, we all observe these thick dark layers. And given that the age control is pretty good here, we're sure that they were deposited under glacial sea level low stands. And we also see these dark layers in other places, especially marginal seas such as Mediterranean. And these dark layers are known as sapropos, and they all interpreted as results of strong ocean stratification. So the dominant theory is that there was this lack of saline water inputs in the surface layer is replaced by a fresher water, and this difference in the density make the ocean, it's like a gigantic lake, and this doesn't ventilate, and slowly the oxygen in the deeper zone is exhausted. Yeah.
Great. Yeah, thank you, May. And one question from online, and one general question. Nicholas is asking, instead of climatic impact on biodiversity, biodiversity impact of climate. So he asks, does increasing biodiversity into the ocean could help reduce the oxygenation or acidity? Is there any publication example? You have any? How?
So
sorry.
So in the world ocean, I guess it would be interesting to think outside of the Sea of Japan. So I can think of the biological pump, that's the sea surface productivity, or the phytoplankton in the surface that sequester the CO2 through photosynthesis, and that would indirectly mitigate the deoxygenation or acidity we see. And yeah, I'm not, it's so I'm not sure it's the richness of species, like if more species, then we see better deoxygenation. I'm not sure about that.
Yeah, true.
Yeah, it's not a linear relationship.
Yeah. So one more quick question from Mark Costello. He said, May, very nice talk. And he's asking about carbon content. Does sediment carbon content increasing during anoxic event due to lack of benthic bioturbation, but continued input from plankton? Or does the absence of benthos simply mean less carbon in sediments?
So the dark layers I mentioned in this talk, for sedimentologists that have looked deeper into the fabric and the contents of each dark layer, show that actually each dark layers are not identical to each other, and they generally indicate there was an anaerobic condition in the bottom water, but each dark layer event was slightly different from each other, only that the thick dark layers show a more consistent content and is more likely all due to the eugenic condition. And in this case, we think that there was very little life in the bottom water here. And so the absence of benthos in your question, absence of benthos mean less carbon in sediments. That's also true. So the calcium carbonate measurement is dropped during these dark layers. And it could be also partly due to dissolution.
Sure, thank you.
Yeah.
So it's about the time. So thank you very much, May. It's a great talk. I appreciate, and I'm excited to know your recent research. And let's move on to the second half. And I'm really glad to introduce Professor Curtis Deutsch. And he's a professor in Princeton, and he's interested in many different topics, from biology to oceanography, into shallow time paleoceanography and deep time paleobiology. And some of his particular interest is on temperature and oxygen and their impact on distribution, body size, and biodiversity via metabolic theory. So he used both mathematical modeling and also empirical data to compare each other to better understand metabolic theory, how it affects distribution and other biotic metrics of marine ecosystem. So yeah, I'm very excited to have Curtis here, and let's start. Curtis, may I ask you to start?
Yes, thank you, Moriaki, for the invitation and introduction. I assume you can see my screen now, so I'll go ahead and start. I'm really happy to be following May because she set a very big picture background for what motivates much of my work. And today I'm going to talk about a particular paper that just came out last week in which we're using a simple kind of metabolic framework for understanding how body size responds to changes in temperature. And you can see my collaborators here and my funding acknowledgements. So picking up from May's nice introduction to some aspects of paleoclimate, here's a really beautiful illustration of how climate change in Earth history has altered the size of marine animals. In this case, it's the ostracods from the seafloor, and they show a really profound anti-correlation with temperature. Let's see if I can get a laser pointer here. So over the Cenozoic cooling, which you see in this blue line, there was a fairly close correspondence with an increase in the size of ostracod fossils. And the question is why? What is it about the climate change that drove animals to get larger, not only for ostracods, but for in many other species as well? And so to address that, I'm going to turn to something more recent, which is laboratory experiments that have been conducted over the past several decades. And those are nicely compiled and illustrated here in a paper by Forster et al., in which they've collected the change in mass per degree of ambient temperature from experiments in which organisms are reared, taken out of nature and reared in a lab under a range of temperatures. And that's been done across animal species that span a very wide range, many orders of magnitude in body size. And you can see that for the most part, most of them fall below zero. That is to say that the change in mass per degree of temperature change is negative. They tend to mature at smaller sizes as waters get warmer. It's not always true, but it's predominantly true. And there's also a hint that, at least among the marine taxa, that size reduction tends to get a little bit greater as you move up the size scale. So these are interesting observations. It's been hypothesized in this paper and elsewhere that the fundamental mechanism driving this pattern is the limitation of growth by oxygen. But it raises a number of questions. Can we kind of quantitatively account for the magnitude and variability in this pattern? Would it persist up to larger sizes in the body size? And can we actually justify or explain this pattern based on oxygen limitation? So to do that, I'm going to turn to a framework that my group has been developing over the past several years, termed the Metabolic Index. And if you haven't seen it before, the Metabolic Index is conceptually quite simple. It's a little bit technical and has several bits of notation, so I apologize for that, but you're going to see these several times. And so I wanted to start off with an explication of this. The Metabolic Index in its concept is nothing other than the potential supply of oxygen that an organism could extract from the environment. That's a rate S, divided by the resting rate of demand. So it's a supply and demand concept as a ratio, where the supply can be a function of temperature, and it can also be a function of the oxygen ambient in seawater. Obviously, if there's no oxygen, there's no supply, and supply will increase with oxygen. The demand can likewise be a function of temperature, the famous temperature dependence of metabolism that's been known for nearly a century. And it can also be a function of oxygen if oxygen becomes limiting of the rate of demand. So from some fairly simple principles, we can write down some simple equations that empirically describe fairly well the rates of supply and demand. And when we take the ratio, we get an equation that looks like this. So we start off with a coefficient called A naught here. And you can think of that as the hypoxia tolerance. It's actually a ratio of a supply coefficient to a metabolic demand rate or a resting metabolic rate. But it measures the tolerance to low oxygen conditions that can be modulated by the change in body size. So body size B can influence both the supply rate and the demand in fairly well-known ways. The body size can be raised to the power epsilon, and that's going to be a really critical parameter for us today. That is the difference between the rate at which oxygen supply increases with size. So if you like, think about something like the size of the gill that increases with the size of the body. But it also changes with the metabolic rate of the organism. We're going to refer to that as delta. So sigma and delta, supply and demand, those two allometric exponents get differenced to give us the body size dependence of the oxygen supply to demand ratio. Or to put it another way, it's the allometric exponent for the hypoxia tolerance. So if epsilon is 0, it means that hypoxia tolerance doesn't change at all with increases in body size. And if epsilon is negative, it means that smaller is better. That is, that things are more hypoxia tolerant when they're smaller, and they lose that tolerance to low oxygen as they get bigger. Then we have a linear relationship to the ambient oxygen in water. So the higher oxygen is, the more potential supply you have. Doesn't mean that an organism will always use that extra oxygen, but it's there to be supplied if rates of demand were to go up. And then finally, we have this term that describes the temperature variation of this supply to demand ratio. And it's written here as a kind of exponential in inverse temperature, which is also known as an Arrhenius function. And the key parameter to focus on here is this temperature sensitivity, E naught. And E naught is just like with epsilon, it's the difference between temperature dependence for metabolic demand or metabolic rate minus the temperature dependence of oxygen supply. So you can think of the famous temperature dependence of demand, well known here. And oxygen supply can also vary with temperature because of things like diffusivity or rates of organismal ventilation. So for the rest of this talk, I really want you to focus on this quantity here, this temperature dependence of the oxygen supply to demand ratio, and this parameter epsilon, the body size dependence. So this metabolic index has been really useful for us in many different ways. Just to highlight a few of them in very brief fashion, we can use it to gain a deeper understanding using organismal dynamic models of organism supply and demand with a lot more physiological complexity, including oxygen binding proteins and multiple steps in the oxygen supply chain. And those tend to, at the end of the day, sort of support the use of a simple equation like this metabolic index. So we can root the fundamental components of the metabolic index in more detailed physiological concepts. It can also work up the tree and give us new insights into biogeographic patterns. We find that the metabolic index goes a long way toward explaining the boundary, at least on the warm, low oxygen side of species ranges, why it is where it is, and how it relates to metabolism. We've been able to show a strong correspondence in certain cases with species migration over the historical record. Here's one from the California current, for the famous collapse of anchovies over the past several decades. And then finally, it was used successfully to predict a geographic pattern, a large-scale latitudinal pattern in the magnitude of extinction at the end of the Permian, the so-called Great Dying, that was confirmed by fossil reconstructions of that extinction magnitude. So the metabolic index is a useful tool. It's rooted in physiology, but gives us insights into all kinds of climatic and ecological processes. But you'll see notably missing from this anything about body size. So I'm going to turn to body size now and see if the metabolic index can help us understand that the patterns of size reductions in response to warming. So here's a simple model. It's both shown graphically up here, but you can write down some simple math to arrive at an equation at the bottom, and we'll do that here really briefly. So temperature on the x-axis, body size on the y-axis. The contours are contours of the metabolic index, phi. So, and they're structured such that low phi values, low oxygen supply to demand ratios are up in the northeast quadrant, and the southwest quadrant are high values of phi. And so if you imagine an organism starting at some body size and temperature at a particular value of its oxygen supply to demand ratio, metabolic index, and some warming happens, the organism finds itself moving toward warmer water. The phi value that it experiences goes down. And if that organism was living at the limit of its oxygen supply to demand ratio, it's going to need to compensate that. And one obvious way to compensate is just simply by making progeny that are smaller over time. And we can calculate the magnitude of how much shrinkage would need to happen to compensate for that amount of warming. And that's a fairly simple algebraic expression. You take the fractional change in phi from the warming and the fractional change in phi from the shrinkage, and you set those equal, and you find that the temperature size effect would be proportional to the temperature sensitivity of the metabolic index divided by the body size sensitivity to it. But of course, not all organisms are living initially at the limit of their oxygen supply to demand ratio. Some of them have an excess of potential oxygen supply that they can accommodate before needing to make any serious taking any serious adaptive measures. So we can split up that change in phi into two components, one that is kind of buffered. We're going to refer to this fraction f, just varies between 0 and 1, as the fraction of reduction in metabolic index that can be tolerated without doing anything to compensate. And then the remainder, 1 minus f, is that which has to be compensated by reducing body size. So that introduces a very simple modification to our earlier expression. The temperature size effect could be calculated if we knew the buffer, the activity buffer, how much reduction in oxygen supply can an organism tolerate before it runs into its limiting value. If we know its temperature dependence of oxygen supply to demand ratio, and then the body size dependence. So these three traits here that could define an organism's physiology and environmental context could be used to calculate its temperature size effect. Now we can measure, or at least infer, all of those traits. The sad fact is that actually we can't get those traits from the same organisms that have been used to measure the temperature size effect. The happy fact is that we've got enough of both that we can make statistical predictions. We can predict the frequency distribution of the temperature size effect from the frequency distributions of each of those individual traits measured on dozens of species. So here's what the frequency distributions of those individual traits look like, starting from that temperature dependence, the temperature sensitivity of the metabolic index, or of hypoxia tolerance, E0. When you use that, divide by Boltzmann's constant and the square of temperature, you get a fractional loss of what you might call aerobic scope, or of the metabolic index, which has a median value of somewhere between 4 and 5% per degree of warming. interestingly, that's roughly in the ballpark of the magnitude of size reductions that we saw in the earlier figure. So we're already just thinking about the temperature effect on the oxygen supply capacity. We are already seeing something that's in the ballpark of the temperature size effect. You'll also notice that it crosses slightly over into negative territory. Some organisms actually increase their oxygen supply to demand ratio as warming proceeds. But the vast majority of them lose oxygen supply capacity or aerobic scope. Metabolic index goes down. In any case, this is only one component. We also have to deal with the aerobic activity buffer, F. Gonna not go into the details there, but we can combine biogeographic data with trait data to estimate that the ratio of the maximum metabolic index that organisms face within their realized niche in the modern ocean relative to the minimum metabolic index that they encounter, to estimate what fraction of their aerobic scope could be lost before they lose habitability in their environments. That has a pretty strong median at around 0.5. So about half of organisms' maximum aerobic scope can be lost before waters become uninhabitable. And both of these have no dependence on body size. In contrast, the third critical parameter here is that allometric scaling exponent, epsilon, which describes, how the body size influences through ontogeny, how the growth of an organism influences its hypoxia tolerance. And here you see that most species are to the left of zero. That is to say that smaller is better. Their hypoxia tolerance decreases as they get larger. There are a couple of exceptions, a few exceptions that are at zero, independent of body size, and one exception on the positive side. You can also see that there's a hint here of a body size dependence. That is, the larger body sizes tend to cluster closer to zero, and the smaller body sizes are further from zero, closer to the smaller is better end of the spectrum. Okay, now we can take all these three traits here, F and E naught and epsilon, put them into this equation and just predict the frequency distribution of the temperature size effect that we would predict without actually knowing what it is. So that is shown in these in the black lines. Here we have using all the traits, the black line, the solid black line shows the predicted frequency distribution of the temperature size effect. The bars show the magnitudes that we get from the actual experimental laboratory data on rearing experiments showing the observed temperature size effect. And you see a very strong correspondence, statistically indistinguishable histograms between the predicted and observed. Now we can do one step better than that, even by breaking up the observed and the predicted temperature size effects into magnitudes that we would predict on the warm side of the median experimental temperature and on the cold side of the median experimental temperature. And what we find is that in both observations and in model predictions, these bars here show the magnitude of the temperature size effect when it's warmer than the median temperature and cooler than the median temperature. And what you find is that the body size shrinkage is a bit greater when the water is already warmer than the median, and it's a bit less when the water is cooler than the median. And that's also predictable from the metabolic index. And it's predictable because we find that the temperature sensitivity of hypoxia tolerance is also greater when waters are relatively warm than it is when waters are relatively cool. That is to say, the aerobic scope or the metabolic index declines more per degree when waters are warm than it does when waters are cool. And that leads to a prediction consistent with observations that the temperature size effect should be stronger in warmer water than it is in cooler water. And again, the model is consistent with observations both under warmer conditions and under cooler conditions. Okay, so that's variation in the body size response to temperature at different temperatures. There's a nonlinearity there, and that's interesting. But there's another interesting feature of the observations that the metabolic index can predict, and that is the variation with body size. So I pointed out earlier that in the compilations of data, people have noted that as body size increases in a log scale here on the x-axis, the magnitude of the temperature size effect gets stronger. That is, that average of something like 4 or 5% shrinkage per degree is smaller at larger body sizes, and it increases as body sizes go up. And it turns out that the metabolic index framework can predict that behavior, as shown in the dashed line, which is the median of some Monte Carlo simulations that propagate the range of variability and uncertainty in parameters. The full variability is shown in the color field, and you can see that it reproduces that tendency for the body size, the fractional shrinkage in larger species to get stronger with size. This you could ask the question, well, why does that happen? It's been pointed out that it does, but there hasn't been an explanation for why. And the answer comes from the fact that that we saw earlier here, that epsilon, that allometric scaling of the oxygen supply to demand ratio, gets closer and closer to 0 as body size increases. And if you just look at the equation that predicts the temperature size effect from the metabolic index, you'll see that epsilon is in the denominator. So as epsilon gets smaller, the temperature size effect gets bigger. In fact, if epsilon goes to truly 0, the temperature size effect will blow up. But now we can look at why does the value of epsilon get towards 0 as body size goes up. And that comes down to, this is my second to last slide, that comes down to the allometric scaling of supply and demand. So here are rates of metabolic rate here in red, and proxies for oxygen supply rate in blue. And if you take the derivative of those curves in a log-log scale, you get those allometric exponents. In red, we have delta, and in blue, we have sigma. And you see that there's a big difference between them. The allometric scaling of demand is much greater than for supply at small size, and they converge toward the same value, something like 3/4 at large size. And that means that epsilon, which is the difference, starts off strongly negative at small size and increases toward 0 at large size. And we can see that in the observations, in the dots, and we can also construct a simple theoretical model using diffusive boundary layer scaling that reproduces that behavior. That is the underlying reason, I think, why the temperature size effect increases with size. And what that means, coming back to the very beginning in terms of climate, is that we take the temperature size effect, we can project using both temperature and oxygen what we might expect the fractional body size change to be by the end of the century. And we get something like a fairly significant 20 to 30% on average at the relatively small body size. That, of course, depends strongly on the temperature sensitivity of species, which is highly variable. It also depends strongly on that allometric exponent, which is also highly variable. So with that, I think I'll just leave you with my conclusions, which are that the metabolic index provides a mechanistic and quantitative prediction of the temperature size rule. It appears to be consistent with direct observations and with the measured physiological traits from laboratory experiments, and that supports oxygen limitation as a driving cause. The convergence of those allometric exponents for supply and demand leads to the prediction supporting observations that as body size increases, the fractional loss of size with further warming gets stronger. Of course, at some point, other constraints will come to bear and prevent further size reductions. One of those may be extinction. And then the differential temperature size effect being larger at larger size may lead to compression of the size spectrum of the marine food webs, which would further amplify what is happening from human harvesting of marine food webs. And I think I'll just leave you with the publication. Again, it just came out last week. If you'd like to find more details than I could fit in a 20-minute presentation. I'm happy to take your questions, and thanks again to the GO2NE Network and Moriaki for the invitation.
Yeah, thank you. Thank you, Curtis. It's fantastic talk. I really excited to hear your new research. And I'm invited May to give a first comment or question, and then I pick Q&A from Q&A box. So please type in if you have any question.
Okay. Thank you, Moriaki, and thank you, Curtis, for this really cool talk, and on your congratulations on your new paper. So just a general question. I was wondering, you've worked on like modern experimental data and work with fossil record across Permian-Triassic extinction. And do you have any thoughts on how this, when looking at the changes in body size or this extinction at different time scales, do you have any thought on how this would scale to at different geological time and scale, and versus what we see in the next couple 100 years?
Yeah, great question. I don't have any results to share on that question, but I have speculations. My guess would be that, over longer time scales, the ability of traits, either E naught or epsilon, for example, to slowly adapt to changes in the oxythermal environment. And that might, on longer time scales, lead to a reduction in the temperature size effect. So that's one kind of hypothetical expectation. On the other hand, if we were to just compare temperature and body size in the paleo record, we have other things besides temperature that are acting to probably amplify the response of body size. So, for example, sudden warmings at least tend to be associated with a reduction in a net primary productivity. So food supply could be reduced. Also, that tends, as you've nicely illustrated for the Sea of Japan, affect the oxygenation of deep water. And to the degree that warming is associated with a reduction in oxygen, not in the Sea of Japan, but elsewhere, then we would expect that to also amplify the direct thermal effect on body size. So there's, I think, over those timescales where we start to bring in multiple factors, and including adaptation and multiple environmental drivers, I think the situation is going to get much more complex and also interesting. So stay tuned. We will be working on that in the coming years.
Great. Thank you, May and Curtis. And there are two related questions. One is from Mark Costello and the other from Mike Roman. And Mark is asking how similar or how different between your model and Daniel Pauly's gill size hypothesis. And Mike Roman is asking, you say the different animals show different sensitivity. So that means larger animals tend to have gill, and smaller animals tend not to have gill and directly get oxygen from surface. It may be a part of reason. I think you can answer both of two together.
Yes, thanks. Those are great questions. So there are some consistencies between this work and the gill oxygen limitation theory. I think that one important difference is that we come to a similar conclusion, which is that oxygen supply allometry in this red curve exceeds the oxygen supply allometry, which is the blue curve, and with observations in the respective points. I think that there's an additional support for that comes from this work, which is the direct measurement of body size dependent hypoxia tolerance as determined by critical oxygen pressure. So I think Daniel Pauly's work would have presented, say, the red point relative to the blue points here, and what we're adding is the green points that say, yes, when you actually measure critical oxygen pressure across size, those support that there being a difference between the red and the blue points. The reason that's important is that, the blue points aren't actually oxygen supply. They're gill area or other kind of morphometric proxies for oxygen supply, which aren't directly supply coefficient itself. So the green data here on epsilon being consistent with that is, I think, a helpful support for oxygen limitation. Now, it's also true that people who have criticized that theory on the grounds that, gills do kind of keep pace with the growth of body size also is consistent with what we're showing. That is that epsilon does converge on 0, which is what physiological criticisms of GO2NE have suggested. So I think that, both sides of that debate have merit, and it sort of depends on, if I were to pick one random species out of this, I would be very hesitant to draw any conclusions. It's really putting them all together where I start to believe that there's a trend here, and it's a trend toward gill oxygen limitation on the smaller size, and the loss of that limitation, meaning gills keeping up with body size on the larger end of the spectrum. Of course, it's that epsilon increasing towards 0 that actually, though, gives the body size response potentially larger magnitude. So that's an interesting twist in the whole thing. And I'm afraid that just answered Mark's question. And I took so long doing it, I've forgotten what the second question was. Could you repeat it, Moriaki?
Yeah, second question is related. So Mike Roman asked the sensitivity difference between larger and smaller animal, because larger animal have gills, but the smaller animal like a plankton, they don't have gill, so they have oxygen directly. So it
yes, right there. Yeah, closely related questions. Good. So this green curve here is a theoretical model that I didn't have time to go into, but it attempts to account in a very simple way for that very transition from body surface area extraction of oxygen into gill surface area extraction. There's a parameter in this model that is the number of exchange elements. So it goes from 1 if you're just a single cell, a unicellular heterotroph with a certain size, and that number of exchange elements grows in a power law fashion as things begin to have gills, where the exchange element is now no longer the body itself, but a single secondary lamellae or something like that. So this green, and it turns out that the green curve has a transition. It predicts a transition in the number of those elements in epsilon that corresponds roughly to where it seems gills tend to start being found in larger sizes. So this theoretical model attempts in some way to account for that, Mike, and I'm happy to talk more about the details of that with you.
Great, sounds fantastic. Thank you very much, Curtis. It's great talk.
Thanks, everyone.
Sorry, it's about the time, so I may not able to pick all the questions, but yeah, hopefully you enjoyed the talk. And yeah, it's about the end of webinar. And yeah, please do not forget, we have monthly webinar. So next one, we are working hard for August. And one thing I say, the good newsletter is already available, but still it's not ready. We are working hard. So I hope it's later in July this month. And again, thank you very much, May and Curtis. It's fantastic talk. I really enjoyed, and I really glad to catch up with both paleo and metabolic things, thinking about biodiversity and body size. And I really appreciate it. And thank you for everybody to joining us. I hope it's quite fun webinar we all enjoyed. And thank you very much. And have a good day, have a good morning, and have a good evening. See you.